MIT Technology Review: Biological teleporter could seed life through galaxy. Starting with just a digital file, scientists manufactured the common flu virus. By Brian Alexander, August 2nd, 2017. The first biological teleporter sits in a lab on the lower level of San Diego building that houses Synthetic Genomics Inc., SGI. Looking something like a supersized equipment cart, the device is actually conglomeration of small machines and lab robots linked to each other to form one big machine. But this one can do something unprecedented. It can use transmitted digital codes to print viruses. In a series of experiments accumulating last year, SGI scientists used genetic instructions sent to the device from elsewhere in the building to automatically manufacture the DNA of the common flu virus. They also produced a functional bacteriophage, a virus that infects bacterial cells. Although that wasn't the first time anyone had made a virus from DNA parts, it was the first time it was done automatically without human hands. The device is called a digital to biological converter, was unveiled in May. Though still a prototype, instruments like it could one day broadcast biological information from sites of a disease outbreak to vaccine manufacturers or print out on-demand personalized medicine at patients' bedsides. We have been dreaming for about a decade of the ability to fax life forms, says Juan Enriquez, an executive with Excel Ventures, a venture capital firm that has invested in SGI, who imagines a new industrial revolution with the digital biological converter as the cotton gin. Craig Venter, the renegade biologist who founded Synthetic Genomics in 2005, but no longer takes a day-to-day role in its activities, has said he even thinks it will be possible to transmit life forms between planets. He's talked to Elon Musk about it, says Dan Gibson, SGI's vice president for DNA technology. Flu viruses, unlike Venter, known for boasts and big scientific plans, Gibson is understated, although also known amongst biologists for Gibson assembly, a reaction that joins small pieces of lab-made DNA into larger genes. SGI Bio XP3 2000, a 3200, a commercial DNA printer forms the heart of the digital to biological converter. When Gibson, sitting in his office, sends a message to the converter, it begins its work using preloaded chemicals. He could just as easily send such a message from anywhere. In late May, Gibson's team disclosed how they'd use the device to create DNA, RNA, proteins, and viruses in an automated fashion from digital transmitted DNA sequences without human intervention. Work on the converter began around 2013 when SGI and Novartis, the drug maker, ran a test to see if they could use data from flu outbreaks to very quickly construct seed viruses from which vaccines are made. Their chance came in March on the year when Chinese authorities reported H7N9 flu infections and posted the bug's DNA sequence data online. The H and the N in flu types refer to hemagglutinin and neuraminidase, proteins on the outer shell of the viruses that are recognizable by the human immune system. It was Easter Sunday, Gibson recalls, when I got an email that H7N9 bird flu was causing quite a scare in China. So we were very quickly able to get the DNA sequence. 2 days later, without direct access to any specimens, only the digitized sequences, SGI had synthesized the H and the N genes on Gibson's DNA printer. Those DNA strands were shipped to Novartis, which used them to generate virus stocks containing the new genetic information, the kind used in vaccine production. Gibson says that when the idea for the digital to biological converter became real, I said, "Can't we integrate everything into one box?" he recalls. Gibson hopes to turn the device into a profitable business. Imagine, he says, that the Centers for Disease Control and Prevention in Atlanta unravels the genetic instructions for an antibody to a disease like Ebola threatening to create an epidemic. That code could be streamed digitally to converters at every hospital around the world to start making the antidote. That's something I believe is going to happen not in the far future. Error problems. Even though the ability to program life and have it appear in different places is startling, just how useful biological teleportation will be remains debatable. Creating a small amount of seed viruses stock is important, but it is only one step in manufacturing enough vaccine for a whole country. The weakened viruses that go into flu shots usually have to be grown by the trillions in chicken eggs, a carefully planned process that takes half a year, and the DNA strands made by SGI's converter will suffer from errors and random mutations. This mutation rate would be unacceptably high if you were trying to make vaccines or pharmaceuticals. University of Alberta virologist David Evans said in an email that FDI would have a cow. Even so, Evans says that although there's not much novelty in any particular step, putting them all together to make functional DNA come out at the back end is quite impressive. Fix the error problem and you'll have a device everyone would want. Reduce bottlenecks. Gibson says he is attacking the error rate problem and is also trying to shrink the converter to a manageable size. Right now, it occupies about the same space as a Fiat 500 car. panspermia. SGI is not printed life just yet. Most biologists do not regard a virus as being alive, but they might get there. In 2016, SGI has announced the creation of a minimal cell, a bacterium with the smallest ever genome, and which could serve as a kind of blank cassette to accept new genetic instructions. Gibson says that once the minimal cell is the simplest form of life, it might be logical to try to print one. Some of SGI's backers, including Venture, are hinting at the ultimate job for a life printer would be to send life back and forth between planets. In one scenario, a sequencing machine could be sent to Mars to obtain the genetic code of any life forms or near life forms found there. The data could then be transmitted to the earthbound converter, which would construct the alien life. Perhaps a high-quality security lab and SGI team spent time with NASA scientists in the barren Mojave Desert in 2013 testing aspects of the theory. We loaded up a research lab bus with all the stuff we needed, isolated some samples, and sequenced them, Gibson says. Sending life data into space could be more interesting. According to one theory of how life emerged on Earth, known as panspermia, it was carried here on a meteor or comet. Sending biological converters into space could be mankind's way of returning the favor, Enrique said, seeding life elsewhere. I want to do something out of this world, to seed one of these things to Mars and print fuels or print part of an atmosphere or nutrients, he says. This invites the question of life form, of what life form or what genetic sequence would be first to go. Venture famously and secretly used his own DNA as the basis for the first human genome sequence of an individual person, published in 2003. When asked if Venture now intended to reproduce his genome on another planet, both Gibson and Enrique said the same thing. No comment. By Brian Alexander. Venture and Musk discussed this agenda. This is just insane to match. Animals gotta pay. Honestly, I can't believe I'm reading this. This is the world we're building. The BlackRock, CIA, telecom, blockchain, mixed reality, horror show of peak capitalist Wetiko, Brookings education, content, Figure 1, giraffes can pay for their own protection. For better stewardship, often... Look at this. Unbelievable. For better stewardship, often this will be undertaken by very poor communities. Herders might be compensated for moving their cattle away from the giraffes. Villagers might earn L marks from planting trees, building fences, and keeping water holes in good shape in drought conditions. Payments will be made for accurate sightings of giraffes and for observation of spoor prints and hair. The animals will pay for periodic drone and satellite imagery, weather and farming data, and economic and security intelligence, internet connectivity from Provid data, and economic and security intelligence internet connectivity from providers such as Starlink may be paid by the animals in LM. They may also pay to protect other related species such as bees and other pollinators and the oxypecker birds that protect them clean of insects and infections. A Nubian giraffe may pay for its very own veterinary care with LM. Since translocations of giraffes are well established, it is possible that the herd might finance their own move to a safer location at some future point. Interspecies money. I believe this is a WEF publication. But yeah, this is just insane, so I thought I'd share it. Just goes on and on and on. Yeah, and these plants also need to be held liable. The effects of development in synthetic genomics. 2010 government hearing. Venter and the gang. Committee on Energy and Commerce. Witnesses. This is my last video. See, I writes video, it matches a bunch of my research fairly perfectly. Anyways, peace and love, everybody.